well, thank you again for being here. And the topic today is sex determination and disorders of sex development, which is a vast area. I figure it will take about 6 hours to cover it recently. So what I'm going to do today is to just select a few topics, trying to make a point which hopefully will become clear as I talk. And I should also inform you of the fact that this is mostly literature material. This is an interest of mine, which is mostly a teaching material, because I find the subject so fascinating, and for the medical students or anybody to learn genetics, some aspects of genetics from here is really exciting. I'll just add a couple of personal cases along the way. If we look at this figure from Vesalius' Epitome of the Human Body, the dimorphism which distinguish male and female is quite obvious, and we all know about it. There is no need to use the laser beam to point out the major differences. And so one would think maybe that the sex development proceeds along separate and well-distinguished tracks to end up with such distinguished phenotypes. Right? Wrong. In fact, the sex development starts on the same track, and it separates only later, and then it runs on two parallel tracks. But along the way, there are a number of exchanges, and that should not be used. But if they are, then something is wrong, and we have a problem. And we'll discuss some of these problems. It has been evident and known from antiquity that such mistakes in sex development can happen, as it is clear from this sleeping hermaphrodite in the Louvre Museum. This is an Hellenistic sculpture that dates back about 2,000 years, I guess. Quite obvious. So why sex? Well, primarily, if not exclusively, for reproduction. But why? If reproduction is also possible by in a sexual way, by fission, or by gemation, of course it would be difficult to envision a mammalian complex organism to reproduce in this simple way, but we could think, for instance, of parthenogenesis that we see in bees, that we see, for instance, in this Mexican lizard, which is very similar to these two companions here, but different because it's capable of asexual reproduction by parthenogenesis. So why, during evolution, has sexual reproduction prevailed, in spite of being energetically more expensive? In the case of human, 9 months of effort, and genetically more risky. Why so? Well, let me just briefly give you some numbers on the genetic load that weighs on a human population. 50% of clinically ascertained pregnancies undergoes spontaneous abortion. A chromosome anomaly is present in approximately 2/3 of spontaneous abortions, in 10% of stillborn, and in 3 to 5% of newborn. And this is only for clinically ascertained pregnancy. if we were to include the loss of pregnancies before 6 weeks, which is believed to be high and largely due to genetic causes, we would then come to the conclusion that the vast majority of human conceptuses never make it to postnatal life. And this is largely due to the complexity of the sexual way of doing reproduction. And this is not a new concept. Already in 1889, this German biologist, August Weismann, said that the significance of sexual reproduction cannot be merely that of making it possible the multiplication of individuals, since this can be obtained in other ways, like we said, gemation or fission or parthenogenesis. There must be other reasons. And the fact is that sexual reproduction is genetically advantageous for one, elimination of detrimental mutations from the gametes, two, to ensure genotypic variability among sibs, favoring adaptation to the environment. Diversity is key to the survival of the species. So, let's start from the start. We are not creationists, but even if we were, we could argue somewhat with the idea that God created man, and then afterwards, and this is reminded in this famous French film, after creating man, Dieu créa la femme. God created the female, and I think he did a good thing. So what can we argue about this? We can argue the fact that, to be pedantic, it should have been the other way around. God should have created female first, because in fact the default sexual development is female. And, how do we know that? Well, for a long time, thanks to the work of Alfred Jost, a French pioneer in the study of sexual development, who did a conceptually simple experiment, even though difficult to perform because of the microsurgery which is involved. He took pregnant rabbits, and at the early stage of pregnancy, he was capable of surgically removing the gonads from each fetus, and then replaced the fetuses into the uterus, and when the pregnancy was ready, all the rabbits that were born were phenotypically female. So Jost discovered that the default setting for mammalia embryos is make it female. So let's try to proceed with some order. Sex determination, we can see two aspects, sex determination and sex differentiation. Sex determination proceeds from genetic sex, which is determined by the assortment of the chromosomes, XX or XY, and that the chromosomes decide that whether the sex is going to be testis or ovary. And this passage, the events that take place to go on from the genetic sex to establish the genetic sex are mostly the work of protein products of a number of genes that we'll look at in a moment. But then, when we move on from gonadic sex to the phenotypic sex, which is internal and external genitalia, then here, this stage is mainly regulated by hormones. So here, gene products, here, hormones. One short parenthesis. Gender identity is a different story. Sex is either male or female. Gender identity, as you know, and it's becoming popular nowadays, has a number of variables. And it's determined by hormonal factors. For instance, we know that in the brain there are androgen receptors, and if it makes a difference if testosterone is around or not around. So even before birth, there is some sort of imprinting of the brain by testosterone, which eventually will be concordant with the body image that boys and girls have differently. Then there is education, culture, the environment in general, other factors probably, but if they are, we don't clearly know them yet. So again, sex determination is the togetherness of events leading to the development of the gonad, male or female. It starts with fertilization, which is the mixing of the paternal and maternal chromosomal complement, separation of the germinal line from the somatic line, key moment, migration of the primordial germinal cells to the bipotent gonadal ridge, and finally gonadal differentiation. And as I said before, it's mostly gene products that do this job. Sex differentiation, on the other hand, is the morphogenetic and functional events occurring after gonadogenesis. So once the gonad is established as male or female, leading to the development, male or female, of internal and external genitalia, as well as the secondary sexual characters. And this is under the influence of hormones. All right, so let's take this embryo and describe events that occur between 4 and 6 weeks post-gestation. This is there is a section here of the embryo that is represented in these other panels. And already here we can see the migration of germinal cells from the allantois through the midgut, the hindgut, what is it called? The mesum that bind. Can't remember the anatomical word. The mesenteric, the posterior mesenteric, that allows the migration of the germinal cells from the allantoic space into these gonadal ridges that are going to be colonized by germinal cells. And, okay, the mesenteric hindgut, that's the word. And until this moment, the gonad is indistinguishable in terms of being male or female. So it's still a bipotent gonad, and only later on there starts to be a differentiation of these germinal cells together with the stroma of this region to establish the difference between male gonad and female gonad. just to reinforce the concept that I was trying to make before. The bipotential gonad by itself, left alone, would become an ovary, and therefore a female. For the bipotential gonad to become a testis, there is need of the intervention of a factor. Oops. The SRY gene, which has the capability to deflect this pathway into a different one, forming a testis. And this was very elegantly proven many years ago, already in 1991, when a British team performed this experiment, taking a mouse female early embryo and injecting into it extra copies of the SR1 gene. And when these mice were born, they were male. They were XX males, sterile but males. So indicating that it is not necessarily the entire Y chromosome which determines the sex development towards maleness, but it is this single gene, SRY, although not all by itself. And here it is. This is the bipotential gonad in the mouse. And there are several other genes here that we'll deal with later. Just concentrate on SRY, which leads to the differentiation of the bipotential gonad into a testis. What I would like to point out in this cartoon is the timing. Here at 12 days, 12.5 days post-coitum, we already have one fully differentiated male gonad. While in the case of a female development, the same stage of maturation of the gonad occurs only at 18.5 days post-coitum, which is a couple of days before birth. So why this difference? Well, I think it's rather obvious why, because the male fetus has to protect itself from the influence of the maternal environment, which is female. And it does so by producing its own hormones from very early on. The testosterone from the lining cells, which are interstitial cells, and the Mullerian inhibiting factor from the Sertoli cells inside the seminiferous tubuli. The female has no need, has no hurry to do such early differentiation because it's in a nice, cozy female environment. So, what are the genes? There are many. I'm just pointing out a few. The bipotential gonad is develops thanks to the action of two genes, essentially. Possibly more, but certainly two. WT1 and SF1. WT1 is a DNA binding protein acting as a transcriptional regulator. It owes its name to the fact that the gene was originally discovered as the gene causing the Wilms tumor. a somatic mutation of WT1 cause Wilms tumor. And SF1 is a transcription factor homologous to the steroid hormone receptor, steroidogenesis factor 1. Once we have the bipotential gonad, then the differentiation process starts. If we have SRY, we go towards the testis. SRY is not by itself sufficient. It needs at least SOX9, which is its downstream gene, which then it's the ultimate effector of testis differentiation. What about the female development? We know very little about it. Probably WNT4, we'll see a little more about this, respondin 1, catenin B1. And then there is another gene which probably is involved also in this process of ovary differentiation, which is DAX1. Why do we know that DAX1 is important? Well, because there are cases of, why do we know that if not an inductor of ovary differentiation is at least an inhibitor of testis differentiation? Because there are rare cases of XY fetuses where DAX1 is duplicated on the short arm of the X chromosome. And these cases have sex reversal. So they turn out to be female because a double dose of DAX1 seems to be sufficient to block this path and therefore allow for the default path to operate. Once the testis is formed, as it was evident in the previous slides, we have the Leydig cells producing testosterone, thanks again to the action, the stimulating action of SF1, and the Sertoli cells that do the anti-Mullerian hormone, thanks again to WT1 and SF1. Then the testosterone on one side, with its own androgen receptor, will cause the formation of the internal genitalia in male as male, and the derivative dihydroxytestosterone is responsible for the male external genitalia, while the anti-Mullerian hormone is responsible for the regression of the Mullerian ducts. What I again would like to point out here is the parsimony of the system, in the sense that the same genes are used several times in a different situation, so that even though these are not the only ones, the system tries to minimize the number of genes that are needed to do the job. So SRY is our main character. It's expressed in germinal Sertoli cells. It has only one exon and one H and G box in the protein of 79 base pair with two nuclear localization signals. It's localized in YP113 right here, just adjacent to the pseudo-autosomal region of the Y chromosome, and it encodes a transcription factor of 204 amino acids, again with an H and G box, which assimilates it to a family of similar genes. One of these being SOX9, which is a little longer than SRY, but it shares the same H and G box, which is the critical for the action of this protein in binding to DNA and regulating the expression of other genes. So SRY functions in supporting a cell lineage of the developing gonad whose destiny is to become circular cell. SRY transcripts are expressed for a brief period during early molecular development, which is consistent with the gene initiating rather than maintaining testis differentiation. On the other hand, SOX9 drives circular cell formation and therefore testis differentiation. If SRY is absent or fails to act in time, SOX9 is silenced and development of follicle cells and ovary anxious. Again, the default way, with beta-catenin being one of the crucial components driving the process. And here we have a graphic representation of what I just said. Initially, SOX9 is activated by SF1, but the transcription of the gene is still low. Then it becomes upregulated through the binding of SRY to its promoter, together with SF1. And at this point, the activity of the SOX9 gene becomes higher and sufficient to produce the protein product, which eventually also serves for the maintenance of the activity of the SOX9 gene. So after initiation and upregulation, SOX9 is capable of maintaining its own activity by itself and some help from SF1. And these genes, SRY, the SOX proteins, bind the consensus sequences on DNA, interact with the minor groove, forcing the DNA helix apart and to bend the target DNA up to 90 degrees. So they do a play like this, and this is the way they can regulate the transcription of other genes. As I said before, there is more than SRY and SOX9. In fact, SRY depends on a variant of the Ras-ERK pathway, and which activates SRY and allowing it to operate on SOX9. In the absence of SRY, which is in a female, the same Ras-ERK pathway inhibits SOX9 and activates beta-catenin, which together with other genes, at least FOXL2 and WNT4, will induce the formation of the female gonad. Now, this is you're never going to read this slide, but it's a key slide to me because what it shows is the number of genes that have been involved into the in the processes of sex development, many more than I have mentioned, and really what I would like to point out here is that the only distinctive gene is SRY. All the other ones are either autosomal or X-linked, so are genes that are in common on both boys and girls, males and females. So and they all act to perform the sexual differentiation. And so it's from this, it should be clear, or at least it is clear to me, that when you operate essentially with the same genetic material to do both things, it's easy to get confused and to do those track crossings that will cause the eventually the development of a sexual disorder. Well, this is something we have already covered. Once we have the male gonad, we have the induction of the internal genitalia, the testosterone and the Mullerian inhibiting substance will cause the Mullerian ducts to disappear, to the Wolffian ducts to become epididymis and vas deferens. On the other hand, in the absence of male hormones, the Wolffian ducts degenerate, the Mullerian ducts are induced to form the oviduct, the uterus, and the upper third of the vagina. Likewise, for the external genitalia, it all starts from a undifferentiated anatomy, and then as we move on, available the male hormones, it will differentiate into male genitalia. In the absence of male hormones, we'll have the female genitalia. So let's move to the next portion of the presentation, and let's look at some disorders of sex development, or DSD. The nomenclature has been recently revised in 2008, and all the terms like hermaphrodite or pseudo-hermaphrodite have been dropped, and so used to be called male hermaphrodite is now 46XY DSD. What used to be called the female pseudo-hermaphrodite is now 46XX DSD. The true hermaphrodite is ovotesticular DSD, and so on. And in the same paper, you will find a proposed classification of causes of disorders of sex development. We have the group of the well-known chromosomal disorders, Klinefelter syndrome, Turner syndrome, mixed gonadal dysgenesis, and rare cases of true chimerism, 46XX, 46XY, with different origin of the two cell lines. Then we have a long list of 46XY DSD. It's a long and interesting list, and I shall use a couple of examples from here. Then we have the list of 46XX DSD. It's a shorter list with a rare condition except for one, which is the 21-hydroxylase deficiency, which is the most common DSD known in humans. And some more cases here. So let me first describe a couple of cases of male DSD where there is concordance between genetic sex, which is 46XY, and the gonadic or gonadal sex, which is testis, although it is genetic. And there is discordance between the gonadal sex and the phenotypic sex with female or ambiguous genitalia. So it can be a total sex reversal or ambiguous genitalia. Typical of this category is the Swyer syndrome, where there is a sex reversal with female genitalia, 46XY chromosomes, mixed or pure gonadal dysgenesis. The gonads are usually dysgenetic, so it's almost impossible to tell whether, well, when it is pure gonadal dysgenesis, it's impossible to tell whether it's male or female gonad. If it is mixed, then you may have some areas where you can still recognize a testis, but not enough to allow for a normal male development. So again, the development will proceed through its default path toward female. There is primarily amenorrhea, there is sterility. Those few remnants of testis that may be found in the gonad are likely to be transformed into a gonadoblastoma. So in these cases, there is essentially nothing to do. These females are to be left alone, except for the removal of these streak gonads, this genetic gonads, because of the propensity to form a gonadoblastoma in the presence of the Y chromosome. How does that happen? There are two possible mechanisms. One is in an equal crossing over between X and Y. They do crossing over on the pseudoautosomal region, which is right here. And as I said previously, the SRY is right here, adjacent, immediately below the pseudoautosomal portion. So if the crossing over is not perfect, if there is some level of inequality, it is possible that the SRY gene is translocated into the X chromosome, leaving a X chromosome SRY positive and the Y chromosome SRY negative. If you use this X chromosome in the fertilization, you will end up with a 46XX male because of the presence of the Y, of the SRY. If you use, like in Swyer syndrome, this Y chromosome, which is SRY negative, you will have a 46XY female. This is a rare mechanism. The most common mechanism is by point mutation of SRY and loss of its function. So if you look at these two mechanisms, you would come to the conclusion that all of the cases of Swyer syndrome are sporadic, because if the mutation, well, in the unequal crossing over, it's obvious. It's a sporadic phenomenon occurring in paternal gametogenesis. In this case, if the father were to be carrier of the same mutation, he would have to have the Swyer syndrome himself, so he would not reproduce. This is the rule, but of course there are always exceptions. Like in this case that John Opitz and I included in a review we wrote a number of years ago. I think this family was studied by David Page, but I don't think it was ever published all by itself. And it's a family where the case is Sarah, which is indicated here as a male because she has 46XY chromosome, and she is a case of Swyer syndrome. There is a transversion C2A at nucleotide 248 of SRY with a PT conversion at position 83, which is within the HMG domain of SRY. So what's unusual in this family? Well, by closer inspection, it turns out that this brother of Sarah, 2-2, has scrotal hypospadias. And the father has, if you think it is significant, a left testis which is smaller than the right testis. What is critical here is the fact that the same mutation present in Sarah is present in the father and in this brother. So the expressivity of this mutation is clearly variable within the family. The father is essentially unaffected. Sarah is fully affected. This brother is mildly affected, if you want to. We don't know about these other two boys here. If they are like the father, or if they are maybe from another father, we don't know. The other condition I wanted to talk about among 46XY DSD is the Campomelic syndrome, which is basically a skeletal dysplasia with bowing of the long bones, missing ribs, and Robin sequence. And the cause of this Campomelic syndrome is SOX9 haploinsufficiency. And the skeletal manifestations are due to the fact that SOX9 is a potent activator of the chondrocyte-specific enhancer of the COL2A1 collagen 2 gene. So its haploinsufficiency caused this dysplasia, skeletal dysplasia. In addition to that, there is intellectual delay, and given what we know about the activity of SOX9 in sex development, partial or complete sex reversal is gonadal dysgenesis. So it's a very complex syndrome, oftentimes lethal around the time of birth. Here are two examples taken from the books, and this is clearly a case where there is no sex reversal at all, or even ambiguous genitalia. But this is a case that looks like a girl, and maybe is a girl. However, it could also be a boy with sex reversal due to the mechanism we just described. And this is the skeleton. You will see the bending of the long bones. Look at the femora here, bowed. Again, within the same chapter, although a different mechanism altogether, the androgen insensitivity syndrome, with great phenotypic variability from the Morris syndrome, which is complete feminization. These two girls, probably sisters, have a perfectly normal female phenotype, in spite of having 46XY chromosomes. Their internal genitalia is absent uterus, absent tubae, absent the upper third of the vagina, but they have 2/3 of the vagina present. So they can have a normal sexual life. And interestingly, the diagnosis in these cases can be done, in totally different circumstances. I remember one case many years ago when we made a diagnosis of Morris syndrome on a lady of about 80 years. For some reason, she had not cared about not having cycles, about not having children. She was happy nonetheless, and until she developed the gonadoblastoma, which had to be taken out. In another patient I saw, the diagnosis was made when she was 5 or 6, because she had appendicitis. She had to have appendectomy, and the surgeon, when we operated for the appendectomy, saw this mass, which was clearly a testis, and that was taken out, and the diagnosis was made. Then we have some intermediate cases. The worst one is the Reifenstein syndrome, because the ambiguity of the genitalia is severe, and may be difficult to correct. Then we have hypogenitalism in a otherwise male phenotype, and sometimes we have perfectly normal phenotype except for azoospermia. All of this because of mutations on the androgen receptor gene on the long arm of the X chromosome. It's a gene of I can read the number of 8 exons, and the protein contains a ligand binding domain, binds testosterone or dihydrotestosterone, and a DNA binding domains. And the mutations tend to be localized in these 2 domains. Here is the mechanism whereby testosterone enters the target cells, binds to the androgen receptors, goes into the nucleus, binds to the DNA, and regulates the activity of other genes. Interestingly, in the around the 5' end of the gene, there is a CAG repeat and an increase in the number of CAGs. I think there are about 10 or so. If they go up to 40, that will cause the Kennedy disease, which is a mild spinal muscular atrophy, and sometimes the males who are affected also show some signs of incomplete or weak, so to speak, sexual development. And finally, like I said, the most common of the DSD, which is a female DSD, congenital adrenal hyperplasia, well known due to 21-hydroxylase deficiency. It is recognized in 3 different forms. The classical form, which can be with salt waste or with simple virilization of the female fetus, and the non-classical form of late onset. This is the case of ambiguous genitalia in an affected female, and this is the case of a non-classical form. You are supposed to see some sort of hirsutism in this girl and acne, and she will probably have irregular menses as well. The problem is within the steroidogenesis that takes place in the adrenal gland, starting from cholesterol. Cholesterol has 2 main destinies within this process of hormonogenesis. One is to make aldosterone, which saves salt from the kidney, and cortisol, which is the so-called stress hormone, which has vague but important functions. One of these being the going back to the hypophysis and regulating the production of ACTH so that this hormonogenesis doesn't go too fast. The point is that the same enzyme, 21-hydroxylase, works on both lines of production, both of testosterone, of aldosterone, and of cortisol. So if this enzyme doesn't work, the blockade here will induce the upstream products, metabolites, to take a different course, which is normally minimal, but then it becomes substantial, in this case, to produce an excess of testosterone and dihydrotestosterone. So there are, I should have said from the beginning, there are 3 pathways: aldosterone, cortisol, and testosterone. testosterone is minimal. It's not very important, unless there is a blockade of the aldosterone and cortisol production. If this happens in a female fetus, the testosterone and the dihydrotestosterone will induce the formation of male external genitalia, or at least ambiguous genitalia, and possibly salt waste at birth. If it happens in a male fetus, nothing happens before birth, but after birth, the excess of testosterone, normally after birth, the level of testosterone goes down to 0, and it will resume around puberty. But in a case like this, it will remain high from birth to puberty, and this will induce a accelerated growth of the testes, which end up being non-functional, and a precocious puberty, which will end up in short stature because of the foreclosure of the cartilages, of the bone cartilages. This is the gene with 10 exons, and the gene is on the short arm of chromosome 6, in the middle of the HLA cluster of genes, and it is present in two forms. The regular functional gene, it's a called CYP21. It's has to do with the respiratory chain and the cytochrome, the cytochrome protein, and the pseudogene, which contains a number of variants that make it non-functional. And the vicinity of the gene to the pseudogene is important in for our understanding of the mutagenesis of this particular gene, because it occurs mostly through a conversion of the functional gene into a non-functional pseudogene. And this can happen because of an equal crossing over, which can cause deletion of the entire CYP21 gene, or gene conversion by substituting a segment of the gene with a segment of the pseudogene. So in other words, this unequal crossing over can cause the import of mutations from the pseudogene into the functional gene, making both of them non-functional. It can be large conversion, micro conversion. And this is the model we're showing how an unequal crossing over can generate two kinds of chromosome. One, which is essentially a normally functional chromosome because it contains one full copy of the functional gene in addition to the pseudogene and the chimeric gene, which is non-functional. Then the other chromosome, which only contains the non-functional chimeric gene, and so this is a mutant chromosome. Then there are point mutations as well, and there is the possibility of drawing some phenotype, genotype correlations depending on the mutation we have. The condition can be non-classical or simple virilizing or smooth wasting. The same concept is expressed here. Just before I conclude the last case, very briefly, this is one of the rare conditions that is treatable. If you know that the fetus has or is likely to have a congenital adrenal hyperplasia, then you can treat the mother during pregnancy with a prednisone, and that is it essentially cures the occurrence of the ambiguous genitalia, and then it will have to be continued after pregnancy as a treatment for life. If that is done properly, these girls will grow up to be normal women, having children, and so on. So lastly, just one word on a case of rare true hermaphroditism, or as we say today, more in politically correct terms, ovotesticular DSD. This is a boy, basically, who was born with ambiguous genitalia. He has a normal testis, and I think the photograph is the reverse because it's on the left. He seems to be on the right. But on this side, he has a normal testis. On this side, he has this hernia, which was operated on, and inside it, what we found were remnants of the uterus, mini salpinx, an ovary, some ovarian tissue with some germinal cells present, still present. 1, 2, 3, 4, 5, and a testicular tissue with some germinal cells as well. Non-functional, too small. The ovary, eventually, these germinal cells without their granulosa will degenerate, so this is totally, it's an ovotestis, totally non-functional. And the karyotype of this in true hermaphroditism or ovotesticular DSD, the karyotype is 46XX in most cases, or 46XX, 46XY. In this case, we have a mosaic with between a minority of cells is 45X, and the majority of 46Y, but in fact the Y chromosome is an intup Y chromosome, and so it underwent a rearrangement, which was probably occurred somatically, and which then generated a second cell line and the phenotype you see. Lastly, the management. Difficult. Once you see a newborn with ambiguous genitalia, days back it was a total surprise before amniocentesis and ultrasound. Now there is usually a premonition of it, but not always. In any case, when you see a newborn with ambiguous genitalia, there are two priorities: to be right in assigning the sex and to be quick, which somehow goes against the need occasionally to perform a number of complicated assays. In any case, do not assign a gender, male or female, before an expert evaluation of the case is completed. Expert evaluation requires the reaction of a multidisciplinary team. This is true. Sometimes we call for multidisciplinary teams even if they're not totally necessary. In this case, they are absolutely needed. A gender must eventually be assigned. In every case, you cannot leave a case pending. And doctors must communicate with the parties who have stakes in the case to explain and perhaps to discuss the options. And of course, confidentiality is of the utmost importance. So I hope I came across with a concept that sex development, while ending up in such important different phenotypes, hangs in balance at all times. The genes are essentially the same, and depending on whether they, don't do it right, the timing or the position or the place where they become activated, you can end up with serious problems. Thank you.